Okay, so this is the third video for part 1, chapter 3. We actually stopped at a slide that refers you to a movie, so all I can say at this point is enjoy the movie. And then you have another movie that you're referred to, so that's a double feature for the same ticket. Okay, so some of the junk DNA that we talked about, DNA that is thought to have no meaning or role to play, might be misunderstood or just waiting to be eliminated by a future evolution. It might be waiting to be activated. So when we talk about junk DNA, we have to remember the idea of evolution and the game of millions of years, and be a bit, cautious with how we regard to that. When we look at mRNA, it has the strongest link to unique sequences of DNA, meaning non-repetitive DNA. That means that those sequences are also have the best link to protein coding DNA, right? Okay, you're referred to a movie. So we're talking about non-repetitive DNA as a better indication of protein coding DNA. That's the major point in this part. So it has been found that about 85 to 90% of DNA codes for RNA or protein. A genome of above 1.5 megabases makes the organism already a parasite, presenting the minimum functions needed for a cell to exist. That has been found in other organisms in their genome. Now, it kind of gives us a cutting point, a reference point to where things change. Okay, at that point, when we're talking about a parasite, we're talking about an organism that lives with a connection to a host cell. So by definition, parasites rely on their host cell or organism for metabolic functions. And we find also organisms that are in between prokaryotes and eukaryotes. So diversity is the name of the game. The minimum amount of functions to be an independent organism in the environment is greater than that of a parasite. So you must have at least 1,500 genes to be able to do that. That's more or less what is known now. 25% of the genome are actually conserved genes, meaning that theoretically they are essential in some kind of way because they have been conserved throughout evolution. And yet they cannot be related to a specific function. So they have something to do with the variety of functions and the ability to function as an organism. So when we're studying unique parts of the genome, one of interest or sequences of interest, right? Components of interest would be those that cause diseases. And basically they'll be used to improve any kind of medical treatment. So we find what we call pathogenicity islands. Pathogenicity islands are actually components that generate diseases. And that is one of the major pillars of the drug and medication industry. So the number of different types of genes is lesser than the total number of genes. That has to be logical, because you take genes and you assume that you're going to group them. You're going to have less groups than the total number of genes in themselves. So now we're going to talk about grouping genes in a way. So when we look at a group of genes and we look at their members, what we call families, they are usually linked by a similar or identical axon or exons. So they have some kind of component that is a basis for all of them. By definition, a family of genes has at least two genes. So theoretically, the number of genes in the body are the number of genes that are associated with families and those that are unique. That's just basic logic when you think about it. So a question that can be brought up and discussed is the question of, is the proteome the exact outcome of the total number of genes? Meaning, are all genes expressed through proteins? So you see on the next slide that you should know by now that the answer is of course not, right? The proteome is larger than the total number of genes because of two reasons. One, we have gene duplication, right? And the second is the other process that we talked about called alternative splicing. So you know that we can have more proteins than genes. All right, let's discuss the term orthologs. Orthologs are genes that code for corresponding proteins in different organisms. So basically, two genes are orthologs if they have about 80% of similar length and sequence. Okay, and you have a movie, so enjoy it. The proteome of a cell can be used to identify genes. So one way to do it is to use protein mass. Okay, so you have a certain mass of a certain kind of protein in the cell, and you try and reverse the process and understand how you can identify genes through that. So we can tag certain proteins in certain areas. We're not going to go into them, but basically we're going to use cDNA expression. Okay, and it's also used, this tagging is used to compare between two different kind of tissues in regard to protein mass. So proteins interact with each other in different ways. One that we've already spoke about was protein complexes, right? And the other one is transiently between them. So this gives us, and the arrays of tagging proteins give us a glimpse of the total number of possible interactions, uh, complexes, and pathways of a cell. So basically, it's going the other way. We're taking a cell, we're tagging proteins, and we're kind of stalking them. We're kind of, walking after them and seeing, kind of conducting surveillance of the proteins. So we know the amount, we know how they interact, we know in what pathways they're going to work, we know in what complexes they take part, and by that we can start understanding more about the cell and more about gene expression. Does that make sense? Alright, and now we're going to talk about pseudogenes. Pseudogenes are copies of genes that have become non-functional because of what we call inactivity mutations, meaning mutations that make genes inactive, right? The rate is between pseudogenes to active genes is about 10 to 1, meaning that there's 10 times more active genes than pseudogenes. Right? You're referred to a movie. Okay. So now we have a slide with a question, and I find this question one of the most interesting questions there is. Is the amount of how is the amount of genes determined? You might say it's a stupid question, but if you think about it, you're going to find out that it's a very smart question. So here's the thing. Let's say theoretically that the cell has a certain size. Does that determine the amount of protein and the amount of DNA and organelles that can be that can exist within the cell? So is evolution making sure that it can fit into the cell, or does the cell change according to functional need? If you know the answer, I'd love to hear it, because I'm not sure there is a certain answer. And of course there's the third option, something in between, right? But theoretically, those are two options that are very interesting. So in the next slide, we see that we have the number of genes that we don't really know exactly. And if we go to the right, then the number of genes is determined by is dependent upon the cell's functional need or needs. So in this case, cell size is not a limiting factor in evolution. If you go to the left and you say, okay, the number of genes is dependent on the physical space that the cell has, then the number of genes is theoretically limited. If the number of genes is limited theoretically because of insufficient physical cell space, then here's a few questions. Why is there so much junk DNA? Is human evolution space dependent? So those are interesting questions if you ask me. Alright, so if we have a series of certain axons in a gene, does that necessarily mean that the gene is active? And how do we define an active gene? Because here's the question. We're talking all the time, and we must remember all the time that we are talking right now, but the context of the whole course is evolution in a way. And evolution is a game of millions of years, right? So how do we define an active gene? It might not be active now, but in evolution's scope of millions of years, it might become active. So are we going to judge it right now? And I'm assuming there can be a lot of answers to that question, of course. An active gene presents an axon, the first, by the way, that follows a promoter. And the internal axons are flanked by splicing junctions. Okay, so that's kind of a general structure to an active gene. The last axon is followed by a 3' processing by 3' processing signals. Okay, you have a movie, so you can look at that. In addition, an active gene has an initiation codon and a termination codon. During research and with time, a problem identifying the number of axons expressed, not those who exist, those who are expressed, there's a difference, right? Relatively to those in the gene was raised. So there was a problem. It was relatively easy to identify the existence of axons in the gene, but how many of them are expressed? That's a completely different question. And this is due to pseudo genes, different levels of expression, and due to mutation. Alright, we're going to stop here and continue in the next video.